Hello, and we are going to take a look at some of the criteria for writing a strong lab report. So if you're looking at this, you maybe just completed a lab report or you're looking to start one. So these are some criteria that we've come up with, and you can see the headings here for this document kind of give you an idea of the stuff that we want you to be able to include. So up across the top, you've got the aim, the hypothesis, the variables you need to include, apparatus, sorry, it's kind of small text, but I'll zoom in a little bit when we need to. Designing the method, how you're going to collect your raw data in your data collection table in a way that's clear and easy to communicate to others. Data processing, so what you're actually doing with the data, for example, putting it into a graph form. And then probably two of the hardest things to do, the conclusion and the evaluation. So I'm not going to read everything here, but you're aiming for what's in the top row if you're using this particular rubric. So are you identifying a focused research question? Is your hypothesis directly related to the question? And have you explained it with enough detail? For your variables, do you have the correct independent and dependent variable? Usually it's only one independent variable and one dependent variable, and at least 2 or 3 controlled variables, but you should probably try to aim for 3 to 5 controlled variables to really emphasize that you are thinking about all the possible things that could affect the outcome of the experiment. That's really important that you try to do that. Have you chosen all the correct pieces of equipment that you need to use, and have you drawn a nice clear scientific diagram that's labeled that shows all those different bits and pieces? Do you have a method that is clear, and the method that actually is, it can be step by step or it can be in a paragraph form, but it basically means your experiment can be reproduced by somebody else. So your method has to be reproducible. Data collection table means how you are collecting all your numbers and values. So if you do create some kind of table, you should be able to see really clearly what the experiment is. The table should have a title. We'll talk about those things in a second. This is all the good stuff down here we're going to get to in a little bit. What else? Data processing. This is all about the graph. There's so many different things that you have to do to make sure you're making your graph strong. The independent and dependent variable need to be there. Conclusion gives a valid summary based on the correct interpretation of the results. So you have to analyze your graph and look for patterns. You have to try to provide an explanation of the pattern, and wherever possible, you have to try to include some background research. We say it's in the scientific text. Basically, whatever you've done your experiment on, you want to be able to see if other people have done a similar experiment, either in your textbook or online, and then see if their results match yours. So scientists like to share their findings with each other, and then other groups can use those findings to try and come up with new experiments or to help validate their own experiments to make sure that they've actually controlled all the variables properly. And then finally here, the evaluation is where you take a look at your procedure, you evaluate your method and the results you came up with, and you try to figure out, okay, was this a well-designed experiment? Did I control all the different variables that I was supposed to control carefully? Were there other types of errors, random or systematic errors, that I could have done something about to begin with to make my data both more precise and more accurate? If you are wondering what those words mean, there's another video that I made called, I forgot what the title is, Evaluating the Experiment, and there's a good explanation there about the difference between precision and accuracy. Anyways, you're aiming for everything up here at the top. So if you find check marks all over the place down here, it just tells you that you have something that you need to do in order to try to reach your full potential with the lab report. So use the feedback. And the most important thing, let me stop this inking thing for a second, is down here, we're going to take a look at all these little boxes right here. So I'll just zoom in. Hopefully we can see that looks okay. And let's get a red colored pen here. So this is for the aim. And so for the aim here, let's see, I'm going to try to keep this under 10 minutes. So you need to make sure that the variables are correct in your aim. So when looking at the aim, you have to make sure, let's see, we've got the correct variables identified. Everything needs to be clear here. So you need your independent variable and your dependent variable somewhere written into your aim, and it should be short and concise. When you're looking at the hypothesis or your research question, different lab reports will have you do it in different ways, but you could just put hypothesis. You need to give some kind of prediction of what you think is going to happen. What is the relationship between the independent and dependent variable? Try to use some scientific explanation, and if you can, use your textbook. Try to find some information that supports what you're trying to say in your hypothesis. Your experimental results may or may not support your hypothesis, but you've started by using some background information there as well too. For the variables, and this box is actually related to this box right here. For the variables, you need your independent variable, your dependent variable, and a list of controlled variables. And I've asked students to create a table that actually lists these variables, and for the controlled variables, they need to show fair testing. In other words, write a few sentences explaining how you are going to control the controlled variables and why it's necessary for you to do that. Don't forget the units. So if the dependent variable is time, you're measuring time, make sure you mention that the unit's going to be seconds or minutes, whatever you're using. So make sure the variables are correct there. Try to avoid the word amount, and then try to say things like volume, length, time instead. And make sure you've talked about enough controlled variables. Okay, I think that's pretty good. So let's look over at the apparatus over here. A nice scientific diagram showing how you actually carried out your experiment. If you can draw it on the computer, do that. If you're doing it by hand, use a ruler so everything's really clear. Beakers should have nice straight edges. I'm not doing a very good job here. I don't have a ruler for my screen. So have you listed all the bits of equipment that you need? Have you drawn a diagram that explains how all of that is going to work? So make sure that that is actually in there. So let's scoot across here, and now we're looking at the procedure a little bit. Let's see if we can make this a little more thin. Here we go. So for the method, the method is your procedure, and it's the steps that you're going to go through in order to carry out your experiment. You need to make sure it's detailed enough that someone else can actually repeat your experiment, that your experiment is reproducible. If it's difficult for you to do it in paragraph form, then go ahead and do it as a recipe type list, but make sure that you explain, you give emphasis to where it's going to be important that you don't mess up the controlled variables. So you need to make sure that's included in your steps as well. And obviously you need to talk about doing repeat trials. A good science experiment always has repeat trials. That will enhance your precision. And what you want to do is make sure that each repeat trial is carried out in the exact same way the previous trial was. Otherwise, if your data varies too much, you'll have too many outliers, and that is going to mess up your precision. So you want to demonstrate strong precision when carrying out an experiment. Okay, what are we looking at here? This is the data table, and these little boxes are my checklist for a data table. You need a good title that's explanatory. And again, the title here, even for the graph over here, when we get to that in a second, the title needs to be clear. I need to be able to know what the experiment is just by looking at your title. So your title should have the independent variable and the dependent variable in there. I keep writing IV, DV. Units need to be in there. So if you have a data table and you have some things written across the top, trial 1, trial 2, trial 3, whatever these numbers are that are written in here, whatever your numbers or data are, we need to know what these actually are. Are they a measure of time or length? So up at the top, you need to put what it is, time in seconds, blah, 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 blah. It needs to be there. Any of these numbers that go down in the same column need to have the same number of decimal places. So if you're rounding, round to the same number of decimal places. Don't leave unexplained blank spaces in the table. If you couldn't collect data, you need to put a mark in there and explain what that mark is. Try to show the calculations. If the last column is showing average, then underneath, show how you calculated average. Average was calculated by blah, 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 and explain how that all works. Make sure it's very clear. A good tip is if your table is good, everyone should be able to understand what your experiment is just by looking at your table. Okay? Now we get to the graph, those 3 more little bits here. Graph is really important. This should be an entirely separate video, but if you're doing a graph, sorry, really small space here, you need your independent variable down here on the x-axis, your dependent variable. You need the names of what the actual measurement is. You need the units as well too. I'm just marking spaces. Your scale needs to be even. So you need to come up with a scale that makes sense. 5, 10, 15, 20, 25, or 2, 4, 6, 8, 10, or 10, 20, 30, 40, 50. It shouldn't be the individual numbers you wrote in here. So you won't write 24.6, then 25.6, then 25 down here and have them unevenly spaced out. That's going to be a big no-no. And then when you plot, let me get rid of that sad face. Then you have to plot your dots wherever they are, and then you have to draw a line. Now the line, depending on your data, this is kind of tough. You have to decide if it's better to do this to connect the dots, or if it's better to draw a line of best fit going all the way through. So you need to make that decision. And of course, you need a title up at the top. And the title should be clear, like graph showing how the length of the wings affects the time it takes for the device to fall down. See, I've got my independent variable in there and my dependent variable in there. Graph paper, if you're doing it by hand, try to learn how to do it by hand. Then you'll understand what the computer is trying to do once you do it in Excel or whatever other program you have there. If you're using graph paper, try to use most of the graph paper. A teeny tiny graph is going to be hard to be able to figure out trends and patterns. Okay, let's scroll over. Can I move over to this side a little bit? Here we go. And then now we're looking at the last 2, which are the toughest to do well, is writing your conclusion and writing your evaluation. Sorry, my handwriting is really bad. So we wrote up here, we mentioned important things. Valid conclusion, strong explanation, background research should be included, and then for the evaluation that you have to evaluate your procedure. Limitations, weaknesses, errors, improvements have to be suggested as well too to get the maximum mark. So let's see what we have here. So for your conclusion, you need to be able to look at your data and then look back at your hypothesis, refer back to your hypothesis, and discuss your actual values and your graph to talk about if your hypothesis was supported or not supported. Avoid things like I proved my hypothesis, or my hypothesis was right or wrong. The data actually supports or does not support your hypothesis. You are one person or one group that's doing this experiment, and other people are doing this as well too. You're contributing your findings to the scientific community, and your findings will either help to support or not support. Other findings will help to support or not support. And based on that, we can get a big picture of the actual question that's being asked to find out if whatever your independent variable is actually affects the dependent variable. So we gotta be careful with how we say that. Once you come up with your explanation based on your graph, what you wanna do is you wanna try to explain it with some science and make sure that science is accurate, so you're not making up stuff like the anti-gravitational force. So you're talking about gravity pulling things down. Do heavier things fall faster? Try to find information about that and then see if that supports your actual information there. So scientific explanations lacking detail. Research from articles need to be included and cited. So if you do find something online, you don't want to just copy and paste words. That's plagiarism, right? So what you need to do is use that information, explain it in your own words, but say where you got that. So if I decide to say that heavy and light things fall at the same speed if there's no air resistance. Well, where did I find that information? If it's from my textbook, then I write my sentence, and at the end of my sentence, I'll put in parentheses where I got that. Put the name of the author of the book, Jonas. And then the year that book was written, 2011. And at the very end of my lab report, I'll have a references or bibliography section. You'll have to check with your teachers in the library on how to do that properly for the school that you're at. Okay, that's the conclusion. I'm going to move on to the evaluation. This is big. You should be separating your evaluation into two sections. I need some more space down here. So your evaluation should be separated into two sections, and I would call one of these sections limitations, and the other side should be suggestions. And it's good when you make it a table like this, because for every limitation that you write, immediately next to it, you can write what the suggestion is. You don't want to say what went wrong with your experiment and then not give a proposal for how you could actually improve it. So let's see what we have here. For the evaluation, I could say you need to talk about problems with the plan. So did you not control your controlled variables very well? Were you dropping things from different heights each time? Was there a controlled variable that you forgot to discuss? Should you be discussing additional controlled variables? Whoops. So you should put that as a limitation. One aspect of our experiment was that we should have been controlling the height. It was something we didn't control. A suggestion, we should be controlling the height, and we should give a very specific way you're going to do that to make sure that you're not introducing more errors into the experiment when you're repeating it. So more limitations. Sometimes people like to say, my friend didn't time properly, they were goofing around. That's not a real limitation to the plan. That's just you not following directions. So if you want to mention that, you can, but that's not going to be very strong for suggesting future changes for people who will repeat the experiment. You assume that someone who's going to repeat the experiment is going to be following directions carefully. So you don't want to say, oh yeah, and don't play with your phone while you're doing the experiment, and don't talk to your friends about Justin Bieber when you're doing the experiment, and try not to fall asleep. So make sure you get sleep the night before. That's not the plan. Those are just things. You have to assume that everyone's following the plan. So what are the specific limitations in the design or the problems with the plan that need to be discussed? When you suggest an improvement, the improvement needs to be detailed, and it needs to be realistic, and it needs to be something that's really going to actually help, not just try harder. That's not going to be a good suggestion for an improvement. A good thing to do is to always suggest additional trials. So if you only had time to repeat it 3 times, you should suggest that you repeat it more than 3 times, possibly 5 times. Why? Because that's going to help with improving the precision and reliability of the data. So the evaluation needs to suggest ways you can increase the reliability of the data. If you only did the trial once, that's not going to be very reliable. So doing additional trials is going to help that. General, more detail, let's see, more specific. Oh, this is the same thing as up here. That's okay. Discuss how you can improve the precision of your measurements and the accuracy. Those are separate though. Precision by doing more trials and making sure that your control variables are being controlled. Your repeat trials should yield numbers, should produce numbers that are very similar to the last time you made that measurement. If it's totally different, there's some controlled variable that you're having trouble controlling. And that could be random. If it's random, you have set up everything perfectly, but when somebody accidentally opens the door to the room, that introduces a little bit of wind, and now all of a sudden that trial you did was wrong. But if you look at it and you identify the outliers, you can say to yourself, okay, we need to repeat that one because there were some random errors that came in there, but I recognize them, and so I can try to fix them. So talk about what kind of random errors were there and what you would suggest to minimize random errors in the future. Systematic errors are about problems with the instruments you're using to measure. So if you're measuring with a ruler and the ruler is not accurate, I mean, we trust that all rulers are pretty accurate, but there are some low quality rulers out there, so you have to be careful with that and make sure you're not making extra mistakes with some of those little measurements that you're making. So that means if the ruler is wrong, in the sense that the actual divisions are further apart than what an actual millimeter is, then you need to be able to mention that. For the most part, you won't have ruler issues, but sometimes, like if you're using a force meter for measuring forces, then it might have a number on it and it may look like there's already something attached to it based on the reading from the scale, but you know that there's nothing actually attached to it. So that's an example of a systematic error. So overall, in the end, that's one way to look at evaluation. That was a very, very long video in the end, but hopefully you can fast forward through the different parts to see which part you're having trouble with, and it can help you figure out how to do better with your lab report. All right.